what I'm going to be going through today is some recent advancements on our software with respect to dose escalation, which will cover some single agent and as well as some combination of agent models. The agenda for today will start with what is new in the software. Then I will go into a single agent dose escalation and show you what are the advancements with the particular designs and focus on one of the 4 designs that we already have in place for that module. And then I'm going to go into the combination agent dose escalation. I will review the 2 methods that are present in this module, and I will conclude with a demonstration of the software with examples and case studies both for single and double agent escalation. So the setup, just to make sure that we are all on the same page, is in these phase 1 dose escalation trials. Our main goal is to assess the dose toxicity relationship. In first-in-human studies where single-agent dose escalation models are in place, the primary objective is to determine the maximum tolerated dose or the recommended phase 2 dose, and the primary variable is dose-limiting toxicities. On the combination dose-finding studies, which are typically denoted as phase 1b studies, we have the same primary objective, therefore determine the maximum tolerated dose or maximum tolerated contour in some some cases. And we want to do this for either a combination of 2 or more drugs or in addition of a new drug to a registered treatment to increase efficacy. A good design will require a compromise between both clinical and statistical considerations, so I cannot stress enough that the tool that you're going to be seeing in action today is mostly a communication tool. It will help you in making decisions, but mostly it will serve as an agent to communicate results within the clinical team and then make an informed decision for the next recommended dose. So what is new on our software? So we have added different features on the current existing methods. So with single dose escalation models, this is a piece of software that has been available since summer of 2014, where we had included the standard 3+3 design, the continual reassessment methods, the Bayesian logistic regression model, as well as the modified toxicity probability interval approach. So we have made advances in all these fields. So with respect to the modified toxicity probability interval approach, we can display now the trial monitoring table, and we have also added the capability to edit the trial monitoring table. This is, for those of you who were present on the last webinar that we gave with respect to single agent models or have attended trainings that we have given on the subject. This is a new feature on the software. So far, we were able to present this trial monitoring table in when we are actually running the trial within the interim or execution mode of the software. With respect to stopping rules, we have added in all the methods except for the 3+3, where the stopping rules are actually dictated by the algorithm. We have added more flexible stopping criteria. And for example, in the case that you see in your screens, we can accommodate more complex stopping rules by imposing a hierarchical structure with a series of logical operators. We will actually see some of this in action on the live demonstration that I'm going to be running. We have also included accelerated titration, where we can actually start with a single subject cohort and switch to the method of our choice once a dose-limiting toxicity has been observed with these single subject cohorts. And the biggest advancement, I guess, on the software has been to add combination designs. You can see the increased popularity of such designs through the years. And later on, when we actually go through the in particular methods, you will see how the regulators view these type of designs. One thing that I should mention here is that for the software, both for the single and the double agent combination agent approaches, there are two modes for running the software. There is the simulation mode, which will enable you to assess the operating characteristics of your particular design under different reality scenarios. So typically you want to explore scenario when the true maximum tolerated dose is probably on the low end of the dose range, on the middle end of the dose range, or on the high end of the dose range. And besides this simulation mode, we also have the execution mode or the interim monitoring mode, where you're actually going to get to run the trial, enter how many, the number of subjects in your cohort, and then the number of observed toxicities, and then the software will propose the next recommended dose, depending on the method that you have chosen, and that will help you make the informed decision with your clinical team. So let's go into the single agent methods. Again, we have 4 methods available, but I'm going to be focusing on one of them in this demonstration. The background for the single agent methods is that we have a sequence of K doses, D1 up to Dk. Each one has an unknown probability of toxicity, and the higher the dose level, the higher this unknown probability of toxicity is going to be. The goal is to find the maximum tolerated dose, which is defined as the maximum dose for which less than a given proportion of the population will suffer a dose-limiting toxicity. In oncology studies, the proportion is often taken to be between 20 and 35%. So dose escalation designs will approach this maximum tolerated dose via an adaptive allocation procedure, which will either increase or decrease the dose level for the next cohort of patients based on the observed outcomes that we have. So these are the 4 methods that are currently available within the software in terms of single-agent models methods, and we have partitioned them here into methods that utilize the Bayesian framework, methods that utilize models for the dose-toxicity relationship, and methods that utilize probability intervals. Out of these methods, what we will do is, in our demonstration, we will focus on the BLRM, which is a Bayesian model-based approach with 2 unknown parameters, which utilizes probability intervals. The reason why methods like the continual reassessment method, the BLRM, or the MTPI have become more popular up to these days is that because the 3+3 design has shown to have quite a few drawbacks. There have been many presentations and many papers in the literature that will essentially give you a laundry list of what the drawbacks of the 3+3 design is. And the regulators have also taken notice of this and have gone as far as to recommend some of the other methods. So in this FDA guidance for clinical considerations for therapeutic cancer vaccines, you will see that it is noted that the 3+3 design may not be the most suitable approach for gathering information, and methods like such as accelerated titration or continuous reassessment may be considered instead of this design. On the European front of the regulators, they go even further, identifying the typically as referred continual reassessment method, and noting that these methods seem to find the optimum dose quicker, treat more subjects at the optimum dose, and they estimate the optimum dose more accurately. And they go as far as encouraging the use of such methods. So the continual reassessment methods, or the class of these methods, which the Bayesian logistic regression model is part of, utilize the so-called Bayesian framework and Bayesian methodology. The idea behind this is that you try to synthesize initially any external information that you may have available to you a priori. This external information may be in the form of historical trial information, publication, or eliciting expert opinion. These can be synthesized and form the so-called prior distribution, which, along with the likelihood or the observed data, form the updated evidence, hence the posterior distribution. The inference will proceed from either directly this posterior distribution or from functions of this posterior distribution, different quantiles of it, or the posterior predictive distribution. So this is what we're going to base our demonstration of the methods on this Bayesian paradigm and philosophy, which typically seems to be sort of like the way that you naturally tend to think about things. You may have some a priori view on things, then you have data or evidence coming in, and that will help you update your view about the state of nature. So the particular method that I'm going to be focusing on today on the single agent methods is the Bayesian logistic regression model, or the BLRM, which is a result of research conducted by a group of biostatisticians in Novartis headed by Beat Neuschwander. So according to this method, a two-parameter logistic model is used to model the dose-toxicity relationship. The two unknown parameters here are alpha, or log alpha, and beta, or log beta. There is use of a reference dose, which is an arbitrary scaling dose. And then these two parameters also have a natural interpretation. The alpha parameter can be thought of as the odds of a DLT at the reference dose, and the beta parameter can be thought as the increase in the log odds of a DLT when you have a unit increase in the log dose. So for example, doubling the dose will result in an increase in the odds of toxicity by a factor of 2 to the beta. So this model this particular method utilizes this two-parameter dose toxicity relationship, and then instead of specifying a target toxicity, a single target toxicity level of 30 or 33%, you essentially specify a collection of intervals, toxicity intervals, and you're going to make your decision on dose escalation based on these toxicity intervals. So there is going to be the targeted toxicity interval, which will include the standard target level of toxicity, that's 30 or 33%, and there will also be adjacent underdosing intervals and overdosing intervals. So in this case, you can actually have partitioning of those overdosing intervals into excessive or unacceptable toxicity, or you can collapse them into just one interval of overdosing. There are different ways of proceeding to the next recommended dose. One utilizes a purely decision theoretic approach, and the method that is most used, and it is recommended also by the authors, is the so-called escalation with overdose control, in which the primary goal is to try and control the overdosing, try essentially to limit the probability that you fall in either one of these two intervals, and control that. For example, you can put an upper threshold of, say, 25% in this probability of overdosing, and then subject to this constraint, you try to maximize the probability that you fall into the targeted toxicity interval. So this is a constraint optimization problem. Now, there are different issues that arise with this kind of methods. One of the issues that has been criticized a lot is how do you specify the prior distribution? We're talking about two parameters here, alpha and beta, or log alpha and log beta. So obviously, it's not trivial to put in a prior distribution on this parameter space. The prior distribution that is typically used is a bivariate normal distribution, which means that there would need to be 5 parameters specified. If you have a good way of specifying these parameters, then you're far ahead than I am. And that's why what we have done within the software is utilize an indirect method of specifying this prior distribution. And this indirect method depends on eliciting information from your clinical team. The type of information that you may elicit is in terms of either what the best guess for the probability of toxicity of a lower dose and what the MTD is, or the lower dose and the reference dose, or the lower dose and the highest dose. So this is typically information that clinicians are much more comfortable providing to you in this probability space rather than directly on the parameter space. And then we utilize a method proposed by Neuschwander et al., as well as a paper by research biostatisticians at Roche, Hasson and Kinnesley, where they convert this information into a prior distribution. In the previous version of EAST, what you would have to do is you would have to actually run the simulations first and then go into the output and see what the resulting prior distribution is. But now we are offering a prior calculator which enables you instantaneously to see what the conversion between these quantities on the probability space to the parameter space are going to be, as I will also illustrate. In terms of specifying the stopping rules, there is, as I mentioned in the beginning, a collection of different stopping rules that can be utilized. Typically what the authors of the original paper propose is to set the so-called target rule. So this is a rule that says that if the probability of reaching the target toxicity interval is more than 50%, then that will be a reason for you to stop the trial. You can actually have that with any number of subjects, or you can actually impose a minimum on the number of subjects that need to be observed so that this rule takes effect. There are other rules that have to do, for example, with underdosing. In this example, if the highest dose is below the target very often, say 90% of the time or more, then you're essentially in the situation where you're underdosing subjects systematically, and you may want to do something about it, like stop the trial. And then there are different other rules. There is a confidence interval rule, which requires that the credible interval of the probability of a DLT at the maximum tolerated dose is within some range, and an allocation rule, which controls the number of subjects, the minimum number of subjects that need to be allocated to the next recommended dose. And if you select any number of these rules here, you can actually, on the right-hand side, put them into a hierarchical order using and/or operators to see which ones will be the most helpful for you. Once you do this, then there are different types of outputs that you can observe. Whether you are on simulation mode or whether you are in trial execution mode, you will be able to see the options like the interval probabilities by dose, which enables you to see how the dose escalation decision is being made by the software. And in this example, in this case, for example, we are looking at the probabilities of excessive toxicity or overdosing, the probabilities of target toxicity in the green, and the probabilities of underdosing on the blue area. Since our goal is to first try and fix the probability of overdosing to be below 25%, then you can see that automatically these last two dose levels are going to be excluded. So our decision is going to be based on these 4 dose levels, and in particular is going to be based in the one single dose level that will maximize the probability of targeted toxicity. And that particular dose level, as you can see here, is dose level 25, which dose level 40, which among the 5 dose levels that have a toxicity below 25% of overdosing, will have a probability of targeted toxicity, which is the largest, and in this case is 31%. In the type of output that you will see when you're executing the software, which will actually help you also communicate these results to clinicians, includes a stacked version of the same interval probabilities by dose, and also the predictive distribution of the DLTs. This will actually predict, based on where you are with the current model that you have, how many toxicities you're expecting to see in the next cohort of patients if you are using, for example, the recommended dose of 40, or you can actually do the same prediction for any dose level above that. So in this case, for example, you can see that out of the next cohort of 3 subjects, there is about a 67% chance that you will see no toxicities, and there is about a 26% chance that you will see 1 toxicity, less than 10% chance that you will see 2, and a very small chance that you will see 3 toxicities out of 3. So this will also help you make an informed decision. Finally, types of output that you can see, and just to see whether indeed you are doing well in terms of the dose toxicity model that you have considered, is this dose probability graph, which enables you to see what the true model that you have selected is going to be, and what in this orange, what the posterior probability, the Bayesian posterior probability shows you, and so that you can get an idea of how close you are into the model that you have assumed in the case. So this is something that you typically will get both at the simulation mode, but also when you're executing the trial, you can see live how well your data is tracking the model that you're assuming. With respect, so this is the single agent model that I'm going to be utilizing in the demonstration that we will begin shortly. But before we do that, let me also show you, talk to you a little bit about the combination agent models that we have also added into the software. So we have considered 2 methods, as you can see here. They're both Bayesian methods. One utilizes a model for the dose toxicity relationship, and each one of them also utilizes probability intervals like the method that we just saw on the single agent approach. The philosophy behind each one of these methods is that you try to find something that is not as naive as just fixing the dose level for one agent and then try to dose escalate with respect to the other. What you're trying to do is to perform simultaneous dose escalations, when you have 2 agents or more agents, you might have multiple MTDs that exist. There is more prior information that you may also incorporate, but you also have to think about the interaction between the agents, because this type of interaction may be synergistic. So, for example, the 2 agents may contribute in increasing the probability of toxicity than what the 2 dose levels of the agents will do separately. They may be antagonistic in the form that they will actually decrease the probability of toxicity, or there may be no interaction at all. So this is something that, based on a priori information that you might have, you can control in the model specifications, in the design specifications. There is a recent FDA guidance on co-development of 2 or more unmarketed investigational drugs for use in combination, which outline and stress the need for modeling the behavior of 2 or more drugs and their interaction. So based on that, what we have done is that we have included these 2 methods in the software. The first method, which we call the COM to BLRM, has the same core of authors as the original BLRM for single agents, only it extends now to 2 agents. And the second method is a result of the research conducted by the MRC in Cambridge, by Andrew Mander and Michael Sweetning, and can be thought of as an extension probably to the MTPI model, in the sense that it uses products of independent beta probabilities to model the probability of toxicity at each dose combination. Both these methods can be put under a general umbrella, in which the approach that we are taking is to specify an initial dose combination for the first cohort, record the number of toxicities that we have, and then, given a parametric dose toxicity model with priors on these parameters, update our inference to obtain the posterior distribution. Then the inference will proceed from this posterior distribution, and we will choose the next combination based on initially identifying a group of admissible dose combinations, and then among these admissible dose combinations, we use certain decision rules, which differ depending on which of the 2 methods you're using, to choose between them using the posterior distribution. And we continue recruiting subjects until either a fixed sample size is obtained or a stopping rule is satisfied. One of the things that we also need to be careful about with these types of models is that we have to utilize parsimony. We are talking about, we want to use a small number of parameters as possible due to having a small number of tested dose combinations. We have to think about interpretability of the results. So we have to have easily interpretable parameters for a single agent 1 toxicity, for single agent 2 toxicity, and their interaction. And we also have to think about continuity. So if the dose of one compound is 0, then we have to have an automatic simplification of this combination agent to a single agent model. So going into the first method, the combination of the BLRM, this essentially extends the single agent method that we just saw on the previous slides. So we are considering a bivariate logistic model for single agent 1, parameterized by alpha 1 and beta 1. Similar type of dose toxicity relationship for single agent 2, and we also have an interaction represented by a parameter eta. There are different types of parameterizations that you can actually show how you can model the probability of toxicity of a dose combination. The one that you actually see here is what has been presented by a seminal paper by Fall when he was talking about the combination of dose levels, and where you're actually looking at the odds of a DLT instead of the probability of a DLT. So in our case, to ensure interpretation of the parameters, we are working with what Neuschwander et al. Worked with, this relationship here, which essentially models the odds of a DLT at any dose combination as a function of the odds when there is no interaction times a term that involves the interaction, which, as I said before, can be either synergistic, antagonistic, or there could be no interaction at all. So we know about the prior distributions that are considered for each one of these parameters, the alpha 1s and the beta 1s. These are just bivariate normals, just as before. And in terms of the eta parameter, this interaction parameter, a normal distribution is going to be used. And again, there are different ways of specifying this normal distribution, depending on whether it's a synergistic, an antagonistic effect, or whether there is no interaction or no anticipated interaction. Dose escalation will proceed with the same principle, with the escalation for overdose control, where again we try to control the probability of overdosing and subject to that maximize the chance of falling into the targeted toxicity interval. And the types of priors that we use are pretty much identical to what we saw on the single agent case. And again, we have the same prior calculators which will assist you into making this interpretation or this conversion from the probability space to the space of the parameters. There could be, as with the single agent approach, there is a simulation and an execution mode. In the simulation, you will consider different reality scenarios. So there could be scenario where there could be multiple true MTDs, and then the model will, the design will actually pick among them. So the types of displays that you'll be able to see is a display like this, for example, like out of the number of simulations that was conducted, you can see here that 23% of the simulations ended up picking this dose combination of 130 with respect to agent 1 and agent 2. 21% ended up picking this combination, 15% this, and 9.4% ended up picking this combination. When you want to make a decision on which dose level or which dose combination is going to be the one that's going to be recommended for the next phase, you will base that again on the interval probabilities by dose display. Only now we'll have a matrix of such displays. So again, you will want to control the probability of overdosing. So constrain yourself only on the dose combinations that have such a probability below 25% or whatever the threshold is going to be. And then among those dose combinations, you pick the one that has the highest probability of falling into the targeted toxicity interval. So essentially the highest green bar. So with So this is the extension of the BLRM into 2 doses, in the combination of 2 doses. And then the other method that we have considered is the pipe design. So in this particular design, we are targeting a so-called MTD contour, or an MTC. So this is a contour with risk of toxicity for those combinations being a pre-specified target toxicity level. So this MTC is essentially a line which partitions the dose combination space into toxicity probabilities either less or greater than the target. There are, again, each one of the dose combinations that you consider, a posteriori, the probability of toxicity of those combinations can be modeled with a beta distribution. Sorry, a priori can be modeled with a beta distribution. And from the conjugacy of the beta distribution to the binomial, the posterior distribution will also be beta. So based on that posterior distribution, you're going to be able to calculate the posterior probability of toxicity at each dose level easily. And then you'll be able to choose the next dose combination by first selecting a set of admissible doses. So these are doses that are close or adjacent to this line that separates toxic from non-toxic doses, and try to get the ones that are closest to this line. So in a discrete, we have a discrete dose combination space. Πij will represent this unknown true probability of toxicity at each dose level. And once again, we will assume monotonicity. So you'll be increasing the probability, this unknown probability of toxicity, going from left to right on the x-axis, and also going from bottom to top on the y-axis. And also this probability of toxicity will also be increased while you're going diagonally in there. So as an example, if we have when each agent just has 2 dose levels, then there are only 6 such monotonic contour choices. These are the only choices that you see here on the screen. Each contour is represented by a binary matrix, which indicates whether the doses are going to be above or below the contour. So zeros denote probability of toxicity below the target level, ones the probability of toxicity above the target level. So in a general I by J matrix, there will exist I plus J, choose J monotonic such contours. Now, the probability of the toxicity for each one of those dose levels will be a priori modeled with a beta distribution. And the way that the beta distribution will be specified can either be in the parameters, directly with the parameters of the beta. So this is a case which corresponds to a uniform distribution, essentially. This is a beta 1,1 for each one of the agents. So you can see you have that the alpha parameter of the beta distribution as well as the beta parameter of the beta distribution are both 1 for each dose combination. Or you can specify these prior distributions in terms of the probability of a dose-limiting toxicity at each dose combination, as well as the prior sample size. So essentially, what is the weight, how many subjects worth is the weight of this prior distribution. So in this case, we have a case where this beta distribution is based on 2 subjects. This is essentially the sum of the parameters of the beta distribution of the 2. So again, this is a case which corresponds to a uniform distribution, and this is a case that corresponds to a much weaker beta distribution, because the sum of the 2 parameters of the beta for each dose level combination is going to be much smaller than 1. Okay, so this is a situation where you have a weak prior belief, and a priori, all of the dose combinations are considered safe. So once you have your prior distribution, then you dose a new cohort of subjects on the best combination. You record the number of the dose-limiting toxicities, and for each dose combination, you calculate the posterior DLT probability, and you also calculate the probability of being above this target toxicity level, averaged over the contour distribution. And then you use the most likely contour for making the decision. One thing that I should also stress here is that this is a more flexible method in the sense that you're not only constraining yourself into dose combinations, but you could also have different regimens. So you can talk about, for example, dose-day combinations. One agent, the one dimension can be thought as the dose levels, and then the second dimension can be thought of as different types of administration of the drug. So with this approach here, you can actually take care of this type of situation as well. So as we mentioned, we are talking about the monotonicity of these MTB, of these maximum tolerated contours. So we want to use the most likely contour for decision making, and therefore we want to define the set of admissible doses, and then average overall contours for safety constraints, which is the expected probability of exceeding the target toxicity level. When we talk about the admissible doses, we want to define the doses that are going to be closest to the maximum tolerated contour. And we want to be closer from below. Obviously, we do not want to exceed this target toxicity level or this MTC line. And once you do that, then you can select among one of these closest doses. You can have different criteria for skipping doses, which I'm going to be mentioning on the next slide. So you can avoid dose skipping if you want. And if there are multiple doses which are closest to this maximum tolerated contour, then you can choose among them with some sort of a criteria. One of the criteria that we are allowing you to use is the one that corresponds to the smallest sample size, combination of a priori and a posteriori sample size based on this beta distribution that we have used. So I mentioned dose skipping. So dose skipping can either be based on a so-called neighborhood-based approach, where you essentially, those combinations that are allowed to be given are restricted to a single dose higher or lower to the current dose, which in this case is denoted by this black point here. So you can only constrain yourself on whether you have where you are in terms of the current dose, and then just increase one dose up or down or diagonally with respect to that dose. And there is also the non-neighborhood approach, which allows you to consider all of the previous doses that were administered, and then restrict to a single dose higher or lower to any of these previous doses. So, and this is the way that the algorithm will work. So it is now time to go into the demonstration mode just to show you how these methods are used. These are the references that I will leave you and that you'll have available in the slide set that will be made to you available after the end of today's presentation. And what I can do now is go into the software itself just to get an idea of how some of these methods are used. Okay, so this is newest. The dose escalation methods can be found under the designs for discrete or categorical data, and we have all the methods available to you, or the different designs here. So first, going into the single agent Bayesian logistic regression model, I'm going to select that, and you will see that in your screen you'll have 4 different tabs. One, which allows you to set the design parameters. So what is going to be your maximum sample size? And you can actually select multiple maximum sample sizes if you want to essentially try and test under different simulation scenario the robustness of your method, and the cohort size. You can also then select the number of the intervals for toxicity. So the target toxicity interval has to include this target toxicity level, which is typically at 30 or 33%. And you can specify whether you want to be using the escalation with overdose control or the decision theoretic approach, which we haven't covered yet. Then there is the dose skipping options. So in the dose skipping options, as you will see here, there are different cases for not skipping any doses at all, or to the other extreme where you allow skipping of any doses and you don't put any restrictions. You can also put different restrictions in terms of what the dose increment could be, in terms of a percentage of a current dose, on the units of your dose level, or in how many dose levels you can exceed that. The prior calculator is over here. So the prior calculator will allow you to specify or elicit information from clinicians on, for example, what do they think their best guess for the probability of toxicity at the lowest dose level and what their best guess for the MTD is going to be. And then this can be converted directly into the prior distribution. And you can also visualize a chart of the prior confidence intervals, 95% prior critical intervals for each dose for the probability of a DLT at each dose level. So that shows you essentially where you are with respect to the prior distribution. And again, the calculator allows you to specify this prior distribution by either, for example, eliciting quantiles from clinicians in terms of what the chance of the probability of toxicity at a lower dose is going to be and at a higher dose is going to be. And again, by making these criteria more stringent or less stringent, you can translate them to another set of prior parameters on your parameter space. In terms of conducting the posterior inference, this is going to be based on the posterior distribution, which is not analytically tractable. So you have to use some sort of a sampling-based method to obtain samples from the posterior distribution. By default, you can either use the Metropolis-Hastings method, or you can use a method which was developed internally within Cytel, which allows you to use a less computationally intensive method to draw samples from alpha and then from beta given alpha sequentially. So this is the first tab. Then in the second tab, you have your different stopping rules that you can set, and we'll see those for the example that I considered. In the response generation, you get to set your different reality scenario that you want to explore. So you'll have some scenario where the DLT occurs maybe at the middle of your dose range, some scenario where it occurs earlier on, and some scenario where it occurs later on. These scenarios can either be specified manually by what is called the general curve, as the one that you see here, or they can also be specified parametrically by using different types of curves like the Emax curve, which will allow you to add more than one profiles when you want to explore this through simulation. And you can also visualize what those curves look like so that you get an idea, visualize both of them actually, so that you can get an idea of what when do they reach this target toxicity level. And finally, the last tab, which is present in all our simulation-based designs, controls how many simulations you want to run, how often you want to have your results refreshed on the screen, and whether you want to actually save some of your simulation results, either in a format that can be read within EAST or in a format that can be read in your favorite data processing software. So in the particular example that I have already created one here, which I have run 10,000 times, so that we won't have to wait until the software is done. This is typically something that gets run in about 2 minutes, 10,000 simulations on my laptop, which is not the most powerful one, where I am utilizing stopping rules based on the target rule. So if the target toxicity exceeds 80% with at least 6 subjects on that dose level, or if we have reached 12 subjects on the MTD, then either one of these could be used in order to stop the trial. In terms of a response generation, I have 7 different dose levels. I'm using one in the higher end as the reference dose. So this is dose level 50 for the reference dose. And I will be simulating 10,000 replications. I've actually simulated 10,000 replications, and I can give you an idea of what the output looks like. So the output can either be summarized in tabular form that you see here, so that you can get different summary statistics in terms of the average number of subjects that have been exposed at different dose levels, the number of DLTs that have been observed, and the proportion of DLTs. Then you have the MTD analysis. So what was the chosen MTD, and how often each one of the dose levels was chosen as the MTD in these 10,000 simulations. And then you have the so-called dose-wise summary, which enables you to assess where you began with in terms of the true probability of toxicity, how many simulations you ended up trying at each one of the dose levels, and how many subjects you ended up allocating at each dose levels, and how many DLTs you ended up seeing at each one of those levels. And then you can compare those true probabilities of toxicity with, which you see here, with the posterior probability. So you have either the average of the posterior probabilities or using the posterior mean of the parameters in order to get those. And there are different other types of output in terms of the interval-wise summary, so how often you ended up allocating subjects in each one of the three intervals that we considered, targeted overdosing and underdosing, and in terms of cohort-wise summary, how many times you ended up stopping with 3, 6, 9, up to 30 subjects, which was the maximum sample size. In terms of graphical displays, you can see some of those tabular displays in graphical format. So for example, the MTD, which allows you to see how often each dose level was picked as the MTD, and the gold line here represents the true MTD, so that you can see whether or not you did well on your simulations. And you can have a dose probability summary. So this is just to give you an idea of how well your true probability, so this is the blue line here, is traced by the posterior distribution in this particular case. And you'll also have displays about the sample sizes that were used at each cohort, and then these interval probabilities by dose that I mentioned. But these can be either be seen in the simulation mode, so you're going to be see one of them, you can see one of them for each one of the 10,000 simulations, or what is more useful is to see what happens when you're actually running the trial. So once you select your design and you try to run your interim monitoring mode, then you can say, I will enter my interim data. I have no toxicities out of 3 in the first cohort. And then the software is going to propose what the next dose level is going to be. So here we have entered no restrictions. So you can see that the software will propose dose level 40 as the next dose level. And this is a result of this particular decision. So again, what we have to do is we have to constrain ourselves into over-toxicity below 25%, which is these dose levels. And then among these dose levels, we have to select the one that has the highest probability of targeted toxicity. Okay? So this is something that is based on purely the criteria that you set in the beginning. But this is the recommended dose that the software makes, and now this is what you as a statistician will put in place in your trial meetings so that they can make an informed decision. And you can accompany this information by, for example, the predictive distribution of the DLTs, so that you can see that, well, if we indeed go to dose level 40, then out of the cohort of size 3, we have more than 90% chance of seeing no toxicities, and there is less than 10% chance that you will see one toxicity out of 3. And you can actually see that for all the dose levels, the future dose levels that you have. So you can see what that chance is going to be, for example, for dose level 15, okay? Or what the chance is going to be for 0, 1, 2, or 3 toxicities. And again, this decision, the final decision on whether or not to escalate up to a particular dose level will be made by the team. In terms of EAST, you'll be able to override that decision, so you don't have to stick with dose level 40, but you may say, okay, I'm just going to go with dose level 15, since this is what the team decided. I'm going to put 3 or maybe a different number of cohort size on that dose level, and then see how many toxicities I end up observing. And this is the way that this procedure will proceed. In the combination to the BLRM, which is the last thing that I also wanted to show you here, when you have the two agents, the input screens pretty much look the same. So you have the design parameters. You just have to specify different reference doses for each one of the agents. You again have the prior calculator for each one of your two agents. And again, you can specify a normal distribution for your interaction term. So in this case, I'm assuming no interaction. So the mean of eta is going to be 0 with a particular standard deviation that you see here. In terms of the stopping rules, here I'm going to be utilizing all three of them that are available. So I will need to have, in order to stop, I will need to have at least 15 subjects enroll into the trial. My targeted toxicity interval has to be exceeding 50% on the posterior space, and I will need to have at least 6 subjects on a particular dose in order for that to be declared as the MTD. So, and I will use the minimum subjects in the trial as the stopping rule, or the combination of the target and the allocation rule. So you can see how flexible you can be in terms of the stopping rules that you create. And in terms of generating through reality scenario, you can specify reality scenario for each one of the agents, and then this will, the software will convert that into what happens in the two-dimensional space, or you can directly go and fill in this 2x2 table, this two-dimensional table to add what your probability of toxicity for the different reality scenario is for each dose combination. And again, we have the different simulation controls. So in terms of outputs, you will again see the same type of tabular displays, but in terms of graphical displays, you will have now matrices of the number of cases in which each one of the dose levels was declared as the MTD. So you can see here, for example, that the combination of 100 in agent 1 and 30 milligrams in agent 2 was selected more than 20% of the time. And again, once again, you can see how many subjects you ended up exposing at each one of the cohorts, how often you ended up exposing 21, 15, 27 subjects, etc. And when you're actually running the trial, you will get to see also these interval probabilities by dose. So if I begin my situation here at the lowest dose combination level, 100 for agent 1 and 10 for agent 2, and if I have no toxicities out of 3, and I click on OK, then the software will propose the next dose level, which if I maximize here, this display, you will see that it is going to be based on, again, constraining the probability of overdosing below 25%. So here, all of the dose level combinations are below 25%. And then among those, I will select the highest green area. If I were using an unrestricted model, then you will see that the dose level combination that should be picked would be this one. Okay, since I have a 32% chance of entering in the target toxicity interval. However, in this situation, I'm using a restricted dose skipping, so I'm only allowed to skip one dose level to go one dose level up on either direction. So as a result of that, from the dose level 110, which I began with, I can only go up this way, or I can only go up this way. So among the two, the one that has the highest green area or the area of highest targeted toxicity is this dose level combination, and that's why this was chosen. Again, this is just a recommendation of the software based on this very conservative rule that we set. This is something that can be overruled by the clinical team after considering all different types of data, safety data, biomarker data, and so forth, and you can actually override this decision. So with that, I will get you back to the slideshow, and I will be more than glad to entertain any questions that you might have with respect to these methods. Again, the slide set which includes the displays, the different types of displays that you will see in terms of output, graphical, or tabular, will be made available to you at the end of the talk. But please, if you have any questions, I'll be more than happy to entertain them now.
Hello, Pantelis, and our online audience. This is Mike White here at Cytel's Cambridge, Mass headquarters, and just want to thank everyone for being online today. We are now going to move into a question and answer period, and we request that you send your questions through the question and answer panel, and we'll try to get to as many questions as we can with the time remaining, and we will go over the hour to try to answer as many questions and continue with Pantelis. Pantelis, there are a number of questions that are coming now here. We're trying to sort through them. Okay. But one question that has been raised is maybe you could talk a little bit, about decision making when faced with a choice between model-based designs like the BLRM and the BLRM extension that you outlined today, and the model-free designs like the modified TPI or the pipe method that you went through today. So what are the pros and cons in choosing between model-based designs and model-free designs?
Okay. Right. So, yeah, this is a fair question. I think it has been also documented a lot in the literature or in different conferences that compare these types of methods. So typically, one thing that can be said about model-based methods is that they're more flexible, they utilize Bayesian inference, and they use all the data that are available. Contrary to, for example, the 3+3 design that only uses data from the last two cohorts. However, these methods are more complex, obviously, so there is a need for a software that will make at least the statistician's life easier, and then through that, the life of the clinical team easier, and that's what we also try to bring with these methods within EAST. So rule-based methods are simpler to apply, but the way that you would actually choose among them from when you're actually running a trial or you're considering some of these designs is beyond like looking in the literature and seeing what the pros or cons that have been documented, is to actually get to see this yourself. So you have the capability of simulating under each one of these designs, and you can also compare them side by side. So you can compare side by side each one of these simulations and see how well each one of these methods is doing for your particular scenario.
Thank you, Pantelis. And, just looking at the bigger picture, so many times clinicians, and you started by talking about and demonstrated various graphs that can help clinicians and other stakeholders in a trial make knowing decisions. But still, clinicians are often fixed on the old 3+3 methods, while statisticians are moving forward with more advanced methods, especially the Bayesian methods that you outlined today. So in your experience, what can be done to convince clinicians to consider more advanced designs, modern designs, and move beyond the 3+3?
Yeah, so this is a pretty big challenge, trying to make a clinician comfortable, I guess, with the idea that there is a model that would be recommended the next dose level. It is a pretty big challenge. So, and it's one of the key responsibilities of a statistician when he or she is designing and implementing these trials. The truth is that indeed most members of a clinical team will have no or very little experience with such designs, and therefore that's why it is recommended that the main features of the approach will be documented using these visual illustrations, but they also need to be accompanied by non-statistical language and common sense. So for example, with these figures of prior and posterior risk plots, which show the reduction in uncertainty and the change of the shape of this dose toxicity relationship, you need to use simpler language. So instead of using, for example, the posterior probability of a true DLT rate being in an overdose interval is 10%, you might just talk about, the risk of overdose is 10%. And then the other thing, a good preparation also includes and mimicked dose escalation meeting using data scenarios to highlight the various possible outcomes. And this is made available in our case through the simulation tool and through the graphs that you're able to see for each one of the simulations that you run, just to get an idea of what are the different possible outputs and outcomes that you can get.
Pantelis, hi. We have a question from Yun Shen. And Yun Shen asks specifically, I wonder, do we capture additive effects? So I assume that's a question about new EAST 6.4's capabilities. Do we capture additive effect?
Well, the way that you're capturing the effects or the synergy of the effects is through this interaction term. So the model itself is not going to capture the additive effect because you'll have a different logistic regression for each one of the agents. But then any synergy or any antagonism between the two agents will be captured by this eta parameter and by the posterior for that eta parameter.
Pantelis, you mentioned Adrian Mander and the pipe method and employing MTC methods. That reminded me that you and Adrian will be together at PSI Berlin in just a couple of weeks, starting May 23rd, again in Berlin. I know that you're particularly interested in seeing Adrian's talk there. Can you tell us what you're looking forward to and what people at PSI might look forward to in Adrian's talk and to learn more about the pipe method?
Right. So the particular thing that is of interest in Adrian's talk, first of all, it's a nice case study that was actually run within MRC. And this is a situation which illustrates the flexibility of the methods that I mentioned earlier in my talk, where instead of just looking at dose level combinations, you are looking at the combinations of dose levels for the agent that you have and dose administration schedules for those agents. So your two dimensions are not dose, but they're dose and day. And this is something that, for example, the BLRM method lacks. It has other advantages, but it talks to the flexibility of this method, and I'm sure that Adrian will present it in a way to make this more marketable to clinicians and to statisticians. And then we're more than happy to sort of like provide this within EAST.
Thanks, Pantelis. And again, I'll remind everyone that today's slides are available right away from cytel.com/events. And shortly, we'll have the replay available, and we'll email everyone when the replay becomes available. Pantelis, today you focused on a number of the design considerations and a good deal of detail. But can you talk a little bit about each function in support of implementation and actual analyzation of trial results?
So we're talking about the execution mode, I guess, of the software. Is this is this the question? Or?
Execution capabilities, support capabilities, yes.
Right. So as I said, I cannot stress this enough that EAST should be viewed as a communication tool. It's not just a decision-making tool on its own. So when you're actually executing the trial, there is a slew of graphical and tabular displays that will be made available instantaneously to the clinical team, along with which there is going to also be a recommendation for the next dose level. And that recommendation, you'll be able to see how that recommendation is being documented, because there are clear graphs for these, for example, for the intervals of toxicity, and how you're controlling over dosing and how you're trying to maximize targeted toxicity. And then you can also communicate to the clinical team what is going to be the risk of seeing 0, 1, 2, or 3 toxicities, say, for example, in a future cohort of size 3, if you go with either one of the next dose levels, including the one that is recommended by the software. So this is a communication tool. So the decision is going to be made by the clinical team, is going to weigh in all the information, including the purely statistical information that EAST will give you, and then you will have the flexibility within EAST to override that recommendation that the software gives you, and then the software will adapt. This is sort of like the beauty of the Bayesian approach, that based on the data that you have, your software will adapt and you will propose the next methods based on whatever data you have.
Thank you, Pantelis. We're actually addressing a couple of specific questions with attendees right now, but we also wanted to ask you about a couple more specific questions statistically. One thing that's often asked is how sensitive are the models to the choice of prior distribution?
Yeah, this is actually one of the trickiest parts of any model, any Bayesian model, and this is where the power of simulation comes in handy. Obviously, you want to work with whatever type of information you have available to you at the point at the time when you're designing the trial. If you have strong information, then that has to be reflected in your prior distribution, which means that your variances have to be smaller. If, however, you have not so strong information, or you do not want your prior to weigh in more than it usually weighs in the determination of your design, then you can actually make the variability larger. So this is something that is examined through simulation. So you're going to be allowed to play quote unquote with different sizes of variability, and then see how sensitive your results are going to be in the output, in the simulation output that you have. And typically what you want to do is you want to pick a specification that lies somewhere in the middle of these results, that is not too extreme on either end.
Thank you, Pantelis. And we're nearing the end of today's session, but there is a question that we talked earlier about communicating with clinicians and development stakeholders. How would you recommend to help clinicians feel more comfortable with the idea that, and essentially it's a black box that's recommending the next dose? That's no doubt a challenge, a hurdle for many clinicians to understand these new methods. What could you recommend along those lines?
So this is very related to the sort of like the very first point that we made, which is that you have to communicate with visual illustrations, non-statistical language, and common sense. So these being able to talk in non-statistical terms, like maybe get rid of, posterior probability and talk about risk of overdosing, and then preparing yourself as a statistician with a collection of mimicked dose escalation cases using data scenario to highlight the various possible outcomes will certainly help the clinicians. And that's why sort of like simulations are your friend in this case, and we have tried to make it easy to use them.
Thank you, Pantelis. We're going to wrap up today. We will keep this section online a bit to address a couple more specific questions here from our Cambridge office. I would highly recommend that everyone online download the slides and make reference to Pantelis's references in the papers that support the publications that support the new methods, especially the pipe method, which we see a lot of people are interested in, and it is a relatively new method that's emerging and has just been introduced into yeast, and we will continue to modify and to refine the pipe method as developments warrant and as we gain more experience with the method and in the clinical setting. So once again, I'd like to thank Pantelis for his time and expert demonstration today, but especially I want to thank everyone who's online who joined us today and their interest in yeast and the application of these new methods into the clinic. One more time, we will be at PSI in Berlin, May 23 through 25. We would love to engage with you then and talk more about how these methods are changing clinical trial design. Also, of course, at JSM, which is the last week of July into the first week of August in Chicago. Many Cytel speakers with particular expertise in adaptive trials will be making talks in sessions, and of course, we'll have a booth presence there at JSM in Chicago this year. In the meantime, you can always raise questions about yeast, and particularly the new yeast capabilities, by simply going to cytel.com/software-yeast. From there, you'll be able to engage us, find out more about these capabilities, how they're being used, and how they might benefit you. So for today, on behalf of Pantelis Vlachos in Geneva, I'm Mike Weitz here in Cambridge, and I thank you for your attention today, for joining us, and I wish you a very good day.